Hi, welcome to our second video of adaptive immunity. During our first video, we talked about the different categories of adaptive immunity, which rely generally on antigens and antibodies. Remember that the adaptive immune response differs from the innate immune response because it's specific against different invaders. So these invaders are generally they're found by the immune system because of antigens. So an antigen is any molecule that triggers an immune response. These have pretty large molecular weights and they're pretty big, pretty complex molecules that have structures that are unique to the individual. So that means that the body is able to see who that invader is. So that means that, say, E. coli would have a different antigen from something else like a virus or even a different type of bacteria. These don't have to always be large cells, associated with large cells. So these could be different molecules that you're creating antibodies against. So like with allergies, those are mediated by antigens. So these antigens are made up of proteins, polysaccharides, glycoproteins, and glycolipids. So the characteristics there, the antigens have allow the organism's body to distinguish self molecules from foreign invaders. So epitopes are the antigenic determinants, and these are regions of an antigen molecule that stimulate immune responses. So here you can see there's an antigen with an epitope. That epitope is the area that the antibody is going to recognize and bind to. There are also things that are smaller than antigens, and these are called haptens. These are too small to be antigenic themselves, but they can trigger an immune response when they combine with a host macromolecule or a carrier molecule that then creates a complex that the body will recognize as being foreign. But haptens alone might be able to trigger a response. Haptens are usually found in the response with allergic responses, and these would be cosmetics, detergents, industrial chemicals, poison ivy, poison oak, animal dander, things like that. For most allergic individuals, penicillin is able to bind to host proteins, and that's what causes the individual to be allergic to penicillin. So penicillin itself isn't an antigen, it's a hapten. Antibodies are what recognize the antigens. These are immunoglobulins, which are antibodies, and these are different kinds of proteins. This is a defensive gamma globulin that's found in blood plasma, tissue fluids, body secretions, and some membranes of the leukocytes. Antibodies are generally found in monomers. So the monomer means just one, and this is the basic structural unit of an antibody. So they're composed of 4 polypeptide chains linked by disulfide bonds. There are 2 larger heavy chains, which are here. They're the lower portion, the blue portion, and they are pretty long, about 400 amino acids, and they have a hinge region where the antibody is bent. There are also light chains, which are that part that's hanging off that look like cigarettes. There are 2 light chains that are about half as long as the heavy chains, and they have a variable portion on all of the chains. And this is what gives the antibody its uniqueness, so that it's able to recognize the antigen. So that blue part is the constant portion, and that remains the same, whereas the variable V portion, the purple, is variable and recognizes the epitope of the antigen. So that's where the antigen binding site is. This is formed from each of the V regions of both the heavy and light chains on each arm. And again, that attaches to the epitope of the antigen molecules. So the constant region, again, has the same amino acid sequence within one person and will determine the mechanism of the antibody action. So within one person, the constant portion is always constant, and the variable portion would vary based on what the antigen is. So again, here's another more of the antibody structure. You can see the antigen binding site as well as the heavy and light chains with the constant and variable regions. Antibodies have five different classes in humans. Most of them are monomers, so we're going to talk about each of these classes. But here you can see IgG, IgE, IgD are monomers, whereas IgM is a pentamer. IgA is generally found as a monomer, but here it can be a dimer. We're going to talk about each of these different classifications in a second, but the way that I remember what they are is that they spell gamed. So antibody classes are named for the structure of the C or constant region. IgA is a monomer in plasma, but is generally found as a dimer in mucus, saliva, tears, milk, and intestinal secretions. This is one of the only antibodies that's present in milk, so that makes it really important. It prevents pathogen adherence to epithelial tissue and from penetrating the underlying tissues, and this is what provides passive immunity to newborns because it's found in milk. The IgD molecule is a monomer, and it's not really known exactly what it does, but we think that it helps with B cell transmembrane antigen receptors. So it's thought to function in B cell activation by antigens so that it helps to trigger further immune responses. But honestly, we're not really sure what it does. IgE is a monomer, and this is a transmembrane protein that's found on basophils and mast cells. So the IgE monomer stimulates the release of histamine and other chemicals that are mediators of inflammation and allergy. So this allows eosinophils to be attracted to parasitic infections, and it produces immediate hypersensitivity reactions that are found with allergic reactions. IgG is a monomer and is the most abundant class of antibodies within the body because it constitutes about 80% of the circulating antibodies. This monomer is able to cross the placenta to get to the fetus and is secreted in the secondary immune response when you're exposed to an antigen the second time, as well as complement fixation, which we don't need to worry about here. IgM was that large snowflake-looking molecule. It's a pentamer, and it's found in plasma and lymph. And I would say that this is the second most common antibody found within the body. It is secreted during the primary immune response, during agglutination, and again during complement fixation. So IgG and IgM are really the most important, where IgG is secreted when you see an antigen for the second time, whereas IgM is secreted in that first immune response. Antibodies, as you would expect, are just as diverse as antigens are. So the antibody diversity within one person is pretty high. The human immune system is capable of making as many as 1 trillion different antibodies, but they all come from 20,000 different genes in the human body. So the variety of these different proteins, which is pretty amazing, is accomplished by somatic recombination or somatic hypermutation. So during somatic recombination, DNA segments are shuffled, and these form new combinations of sequences to produce antibody genes. Think of them kind of as Legos. You can use Legos to create a house or a fence or whatever, but they're all the same Legos. So somatic hypermutation is where B cells and lymph nodules mutate and they are able to create new sequences. So this would be like buying a new Lego kit or something like that, so that you're adding more diversity to what your body can work with.